Hello, my name is Jean Marie, and I will be presenting the article The Gut Microbiome Modulates the Protective Association Between the Mediterranean Diet and Cardiometabolic Disease Risk. I chose this article because I was searching for something related to autoimmune disease. I know the importance or the impact that this diet can have, and I thought it was interesting to study this connection between the diet, microbiome, and disease. So before going into what this paper did, I want to look a little bit into background to understand their purpose. And there are three factors here. One is the cardiometabolic disease. The cardiovascular disease and type 2 diabetes are within the top contributors of cardiometabolic disease in the US. And another important factor here is the gut microbiome. We know the importance of it, but studies have linked microbial metabolism with risk of cardiometabolic diseases. And then we have the third factor, which is diet. Studies have shown how diet can actually modulate the gut microbiome. And we even saw in the professor's journal club how dietary patterns can have larger impacts in early life. And I'll circle back a little bit into that. But we have seen how the gut microbiome affects what we get in our diet. So there's that connection there. And then the authors talk about this early versus long term dietary patterns and the fact that long term is of greatest interest when studying chronic diseases. And that's kind of what I wanted to circle back to that I found very interesting when looking and when observing the professor's journal club. Now, they point out the lack of long-term dietary measurements in diet microbiome studies. Um, and so that's an important factor that we'll come back to. But there is a two-way relationship here. And again, there are studies that are lacking looking at this two-way relationship. Now, one of the diets often looked at is the Mediterranean diet, which includes higher intakes of fruits, vegetables, nuts, legumes, and olive oil. And then very few, and it's characteristic of this, very few red meats or processed meats. And then refined grains with low to moderate alcohol or wine consumption. Now, studies have shown the benefit of this diet for the prevention of cardiovascular disease and type 2 diabetes. And the authors talk about two studies. There's one that looked at how the med diet modulates the gut microbiome, and studies that have actually shown an increase in Faecalibacterium prausnitzii and a decrease in Rhamnococcus nevus and a decrease in Prevotella species like P. copri. And this one is really interesting, and you'll see why later. But this one has been associated before with insulin resistance. So there is a connection there with type 2 diabetes. Now, this paper wants to look at how all of these three factors are interconnected. And that's where we come with our bottom line up front. So they want to see how the microbiome might modify the interaction between diet and cardiometabolic health. And they have two main goals. And they want to prove and they want to aim to investigate whether the cardiovascular benefits of the med diet vary based on individuals' gut microbial profiles. They want to see and determine if the med diet causes a change in the gut microbiome itself. And they mention that there are studies that the studies are limited by because they're using 16S ribosomal RNA gene sequencing that only yields very general taxonomic profiles like phyla or genera, not species. So they want to, and of course, they're going to be taking advantage of this because it's missing. And then they do, what is it that they find? They found that the protective association of the med diet with cardiovascular disease was significantly stronger in participants that didn't have the Prevotella copri in their gut microbiome. And remember, this bacteria has been associated with insulin resistance. So why is this important? Because this could be that connection between diet, chronic disease, and gut microbiome or microbial profile. And this could lead to dietary recommendations for cardiovascular disease risk patients that can be personalized to their gut microbial profile. But we'll circle back to this at the end of the presentation. Let's start with what is it that they did, and we are going to be starting with their figures. And figure one is actually their experimental strategy, and I really like the figure because they organize very well what is it that they're doing. Now, there is a long running study called the Health Professionals Follow-up Study. And this started in 1986. I find it really cool. They've been doing this with over 50,000 men. I do want to point out these are men, white men, okay? And they do validated food frequency questionnaires every 4 years, and then every 2 years they do medical uses and medical diagnosis, lifestyle, things like that that are updated. A group from this paper, um, there's multiple labs. Well, one of the labs actually did a sub study, and it's called the Men's Lifestyle Validation Study. And they published it in 2018, in which from 307 men they collected from 2011 to 2013, they collected stool samples and a blood fasting blood sample. Okay. Now, from this study, they took that data as well, and they, of course, they already have the diet assessment from this long term study. The stool samples were two pairs of samples collected 6 months apart. Each pair collected from two consecutive bowel movements, 24 to 72 hours apart. And that's where it gives you the numbers. And then the fasting blood samples, I'm sorry, were collected 6 months apart. Okay. Continuing that figure, the Mediterranean diet index was initially developed in 1995 by Willett et al. And this is what they followed: the Mediterranean diet, which participates. They are asked what is it that they're consuming, and some healthy components are fruits, fruit juices, vegetables, whole grains, legumes, nuts, fish, and moderate alcohol consumption. And then a very important unhealthy component is the red meat or processed meat. And the index, their participants' consumption that are below the median were assigned a value of 0, and those whose consumption were at or above the median were assigned a value of 1. Okay, and then with Barry Cordes analysis, we get plots like these. Now, the gut microbiome, they did have, and I want to point out, they had 96 participants only because they excluded those that used antibiotics within 1 year. And it's quite interesting because I was discussing this with other from the cohort, in which the impact that certain antibiotics and also supplements may have, an over-the-counter medication. And we were discussing the fact that these when these studies actually exclude them, they basically focus more on antibiotics and not necessarily supplements or over-the-counter medication. And that's something interesting to be taken under consideration, but because there are studies that show that there is an impact. And we cited a few of these. So these genomes and metatranscriptomes were obtained using Illumina high sequence paired and shotgun sequence platform. The taxonomic profiles were used using meta PHN, and this classifies metagenomic reads to taxa and yields relative abundance in each sample based on 1 million clade-specific marker genes from 17,000 microbial genomes. And they got about 468 species. And then the functional profiles for both metagenomes and metatranscriptomes was obtained by applying human 2.8.0. And you get enzymes and pathways, and then we see them in figures like this that we're going to see in a couple of minutes. And then the cardiometabolic disease biomarkers, they looked at lipid metabolism, like and seeing lipid panel, for example, inflammation, high sensitivity, C-reactive proteins, and then glucose homeostasis. They only looked at hemoglobin A1C. Okay. Now our second figure here, they wanted to look at how the med diet pattern and taxonomic and functional profiles of the gut microbiome. And they first look at the adherence to the med diet. And you can see it here, the med diet index. And some important things here is that you see from the med diet index that was listed in the before, you can see the fruits, legumes. You will have it in a high consumption in individuals that adhere and have a high Mediterranean diet index. Now, looking at red process or red meats or processed meat, you can see that those that do not adhere to the med diet, they consume more of those. Then in panel B, we're looking at the distribution of 10 microbial species, but this accounts of 46% of the community abundance. And the most prominent pattern of gut microbial taxonomic variation is the expected trade-off between bacterial dates like Bacterium uniformis and formicutes like Granulum unclassified. And then the expected, and you can see here, the expected completely or near absent of Prevotella copri. And you can see that there. Now, when we're looking at common housekeeping processes, this is metabolism of carbohydrates, nucleic acids, nucleotides, vitamin biosynthesis, genetic information processing, things like that. The species encoding and transcribing these abundant pathways and enzymes were themselves highly prevalent. So there's this connection that we can see here with the coloring. Now, in the next figure, I did divide it again because it's really large, but they are looking at the med diet adherence that has modest but significant effects on overall microbiome configuration and specific microbial species. So they saw that in this one that in panel A that the med diet was not a driving factor for structural variation in gut microbiota. You don't see a pattern cluster in the med diet index, and they use Curtis dissimilarity as their distance index in this figure, but when they perform a statistical analysis per MANOVA, they actually revealed, and this is here, when they're looking at the proportion of that variation in taxonomy, explained by either the med diet, by cardiometabolic biomarkers, things like that, they did see that its association was significant with respect to both taxonomic and enzymatic structure. So this goes back to what they described in the intro, that long-term dietary intake can exert a gradual selective pressure on the adult gut microbiome, and transcriptional regulatory responses may be influenced by more localized stimuli. Now, they did want to point out that the zero point 0.7 of variation explained by dietary patterns was actually comparable in magnitude to other large-scale investigations. So they did have a comparable value there. In this same figure, looking at another panel, they looked at 40 species, and we see them here, from 4 phyla that were significantly associated with the med index here. So there is a significant association of the med index and its food with microbial species. So in and here we can see that. Now, the med diet driving force of association between overall dietary patterns and microbial features is what's being analyzed here. So a positive association was seen in these 3 species. Okay. Now, F. Praunitzi and E. elegans and Bacteroides cellulosilicus, these are associated, and these had a positive association, but these have been known to be dietary fiber metabolizers and short chain fatty acid, okay, production. Now, there was an inverse association between these, the Clostridium botea, Clostridium pathowai, and then the Collinsella outofasciens. Now, looking at these over here specifically, we can see that there's this negative or inverse association with the Mediterranean diet. And the other ones that I mentioned that had a positive association. But when we look at the red processed meat intake, we can see that these have an inverse association. However, this one has a positive association with the red meat intake. And previously, this bacteria has been linked to Western diets and diet styles, which are high in red meats. And that's why you see this positive correlation here. And then lastly, there was no association with Prevotella copri. Okay. In our next figure, we see here, we're looking at the relationship between med diet to functional potentials of the gut microbial community. So enzymes and metabolic pathways, but these are quantified from the metagenomes. So they did see that there is that it influenced plant polysaccharide degradation and short chain fatty acid production and pectin metabolism. So there was an association between the med diet with 36 pathways and 188 enzymes. There was a positive association with, and these are the most prominent findings, and I'm going to highlight some of the most prominent findings here, were with high metabolic functions for degradation of specific dietary fibers, okay? So in individuals in individuals with higher med diet adherence, they saw that there was a more prominent finding in these microbial functions, which kind of makes sense because plant polysaccharides degradation, the pectin, all of these are typically plant-based, and that's what the med diet is predominantly, plant-based diet. So there was a positive association with a enzymes like these that are associated with degradation of pectin, which is a soluble fiber, which is rich in fruits and some vegetables. So it kind of makes sense to see this positive correlation. We also see a an enzyme here with a positive correlation in the mannan degradation pathway, which is also a major component of the hemicellulose in a cell wall. It's an insoluble fiber, so it makes sense as well to see this upregulated. These are coded by, and they point out here, and you can see it with the coloring, by the bacteria F. Braunsnitzi. Okay. Now, looking at another enzyme here, this one is the 7-alpha dehydroxylase. This removes a hydroxyl group from the primary bile acids, and it's encoded by this bacteria. Okay, and remember, we saw this bacteria as well, and you can see the coloring. So the thing, and you can see a negative correlation here with the med diet index, which we saw before as well with this bacteria, but we can see it with the enzyme. Now, here, this was high in participants with low med diet adherence, and you can see the negative correlation here. Now, the products of this enzyme are well-known secondary bile acids that are actually toxic for the liver at elevated levels. So, and then last pathway to mention here is the lactose and galactose pathway. There was a lower abundance, and it kind of makes sense to that has a negative correlation with the med diet index because it dairy food consumption is low in the med diet. Now these just pointing out, like I said, I was only going to highlight a few, but these are involved in numerous other basic biochemical processes. And here we're looking at DNA copies, which brings us to the following part of this figure. The med diet adherence influences, and we're looking here at pectin degradation, okay? So mainly when we're looking at these bacterias, okay, and remember that pectin is high in fruits, which is high in med diet adherence, okay, you'll see that there. So these bacteria mainly encode and transcribe pectin degradation enzymes. And previous studies have shown dietary pectin degraded by bacteriates, which supports this. But they also saw, which we saw before, F. Braunsnitzi actually contributed to the DNA profiles, but was less active in the transcribing of these enzymes. Okay. So this was an interesting correlation that they saw here. And then the final and the very impactful message that they actually had in this paper was in this figure. And it's to get to here, they were thinking and they wanted to evaluate each participant's cardiometabolic disease risk, but by using levels of biomarkers, like we mentioned in the methodology. So they actually did kind of and designed a separate case control study of 396 myocardial infarction risk patients and from an 843 healthy controls. They did a 10-year follow-up with this. Remember, this is from this original study, which are white males. Okay. So they saw an inversely association between the adherence to the med diet and the cardiometabolic disease risk, which is kind of expected. But to test whether the known association differs in individuals with different microbial profiles, they did a Spearman correlation between PCO1 loading and the P copri abundance. And they saw that this association was more pronounced in participants with a low PCO1 and weaker in participants with a higher PCO1. Now, the PCO1 loading was accounted predominantly by the peak copri abundance, not by other highly abundant species. Now then, the last thing that they did was they quantified the predicted risk of the myocardial infarction associated with the med diet index in carriers and non-carriers. So they saw that there is a 18% lower risk, okay, of myocardial infarction in non-carriers. Now, in carriers, they saw a 30% increase, but it's non-significant in the myocardial risk in P-copri carriers, okay? Now, it's still very powerful evidence because this supports that gut microbial functions and taxa may not only respond to diet, but interact with it, which then modulates resilience to diet-induced cardiometabolic disease risk. And that's part of what their goals were. So as a reminder, their goals were to investigate whether the cardiovascular benefits of the med diet vary based on individuals' gut microbial profiles. And they found that long-term adherence to the med diet was associated with overall gut microbial profiles of phylogenetically diverse organisms carrying pathways for plant-derived polysaccharides degradation, short chain fatty acid production, and secondary bile acid production. And then they wanted to determine if the med diet causes a change in the gut microbiome itself. And they saw that high pectin content, which remember it's fruit derived, may explain the med diet's role in shaping gut microbial function. Now remember, their main question and what we were mentioning at the beginning was how, okay, the microbiome might modify the interaction between diet and cardiometabolic health, all of these three together. And they found that there's a strong protective association between the med diet and the risk of cardiometabolic disease on this subgroup of participants that could be explained by the presence or absence of P. copri in their gut microbiomes. Now, this study, they point out that they had limitations. They had an observational study, which means that it was not possible to determine whether the med diet casually selected for gut microbial features. They did not identify a significant association between the med diet and the abundance or carriage of the P. copri, only the interaction between the diet and the carriage of P. copri with the cardiometabolic disease risk, which was seen nicely in that last figure. So they said this is indirect permit specific speculations. There's two possibilities here. Either individuals who do not carry precopy have gut microbiome that metabolize components of the med diet more efficiently, leading to higher yields of cardioprotective chemical products, or individuals who adhere to this diet are less likely to acquire this strain, and which is then in itself cardioprotective. So I would like to know, what do you think would be the possibility in the discussion? Remember, they want to be able to do a dietary recommendations for cardiovascular disease risk patients that can be personalized based on their gut microbial profiles. So, for example, for carriers for precorporate, do we recommend then physical activity or pharmaceuticals? And then those that are lacking it, just a med diet, for example. Future directions and questions that we can actually start discussing in the discussion or talk about in the discussion. Can the study explain results showing precorporate improve glucose homeostasis? Because they didn't really look much more than A1C. Can they actually do interventional studies that go top down in humans? And we were discussing this in another cohort about looking at maybe then model systems, or maybe we can combine them. Assess both changes in the risk of cardiometabolic diseases in individuals with diverse baseline microbiomes, and then before and after med diet intervention. It will be interesting. And then studying multiple subtypes of pea copper in culture or mice models with alternate plant-derived polysaccharide sources. I do want to point out here that this study was in white males, and they did mention that it was clade A of the subtype. And then, could the microbial biomarkers be used in the clinic? Can they be used for diverse for disease severity or progression? So these are some interesting things that I would hopefully we can discuss in the forum. Thank you so much. These are my references, and I look forward to your discussions.